another lecture series for Geology 3104C at the University of South Florida. This is Sarah Sheffield speaking. So we're going to be talking about ichnology, otherwise known as the study of trace fossils today. And I'm pretty excited for it. So review concepts that'll come into play today. So if you don't remember these from sedimentary petrology, it'll be really helpful if you go back and review your lecture notes. First, how does sediment move? How does it get picked up? How does it get moved? The energy involved within it. Common sedimentary structures like ripple marks, cross-bedding, etc. Where they form in relation to energy of water and wind and things like that. Grain size and sorting, and that relationship to energy. We're not going to go over those directly, but you should remember these concepts because they will come into play with interpreting ichnofossils moving forward. So ichnology. This is a kind of where sedimentology and paleontology meet in the middle. Trace fossils, of course, were made by animals, or living things, so they reflect behavior in living things, but they're also influenced by the sedimentary environment. So they're both sedimentary structures and fossils at the same time. So, for example, on this side here, we can actually see flow directions of these particular trace fossils. On this side here, this is, I believe this is in Morrison, Colorado. It's absolutely beautiful. You will see this very soon in your in-class assignment. So one of the things that we need to worry about is the appearance of an ichnofossil is actually directly controlled by the sedimentation itself. So it's not just due to the shape of the structure, like a dinosaur foot, but it's related to how much water is in the sediment, the grain size of the sediment, etc. So here is an example of how a bird can look on different levels of sedimentation, the depth of which. So depending on where it is exposed and how it is preserved, it can completely change the appearance of that fossil. There's quite a few different categories of trace fossils. We have our tracks, which are discrete footprints, trails, continuous traces, burrows, borings, fecal pellets and strings, and coprolites. So you've seen a lot of these already. Now you know that trace fossils can be left behind multiple times by the same organism. So we can really start to begin to picture these rich behaviors of organisms throughout geologic time, if we're able to interpret them. Now, they're given binomial names, genus and species, just like regular fossils, but they don't correspond to who made them, and that's a really important point. And the reason for that is one animal or one plant can make a bunch of different types of traces, and two, we can never really be sure how or who made that trace. Remember, it's rare for a trace fossil to be preserved next to the body fossil. So as scientists, we can never prove who made it. So we're cautious to name it after the actual organism. So for example, here is Rusophycus. Rusophycus is a resting structure. You can see how it is not really continuous. It's just one little bit. It's a resting structure typically made by arthropods like trilobites, but maybe not always. So here we've got our cute little arthropod here. Laying its belly down into the sediment, making a cute little trace there. All right, so Rusophycus is one of my very favorite trace fossils, and there's a reason for that. All right. Now, again, we rarely know who made it for sure. So here's why this is one of my favorite trace fossils in the whole world. This is in the middle of Clinch Mountain, which is really in the middle of nowhere in Tennessee. And there's a restaurant there called Vinegar Pie. It is not good, Vinegar Pie nor the restaurant, but it's charming. And the reason why this is such a great place is if you go behind the restaurant, where they actually have a dog breeding business right behind this fence. It's a really strange place, folks. It's in the middle of Tennessee, as I said. All right, if you go behind there, here is some of the very best Rusophycus in the entire world. The people who own this restaurant are continuously really weirded out by the number of geologists that come here internationally to look at this trace fossil, which is pretty great. So here we have an example of why we can never name it after a specific fossil, because one animal can make a whole bunch of different traces. So here are just 4 specific traces that this ultra productive crab can make here. We've got a poo trace, We've got a burrow here. We've got all kinds of stuff. Sometimes we can get really lucky and we can see something living today making a trace. So this is one of the great things about trace fossil work, is that we can use living creatures to compare to the past. So Stephanie Drumheller is a professor up at the University of Tennessee and a friend of mine, and some of her research deals with this actualistic taphonomy, right? And actualistic ichnology. So she studies crocodile tooth marks, bite marks in other things. So one of the things she does is she throws cow bones into alligator and crocodile pits, and then she later, after they're done with it, will examine the bones for the bite marks left over. And then she can compare it to the traces left behind in the fossil record, which is pretty cool, right? Super cool research. Also super terrifying, but there you go. So ichnofossils, trace fossils, this is a rule from the International Code of Zoological Nomenclature, the ICZN, the rule book guiding taxonomy and paleontology. Trace fossil names should be based only on morphological features, including shape and ornamentation, and not on who we think the maker or mode of preservation is. So ichnology got a boost. No, it wasn't really a big deal until Adolf Seilacher, he went by Dolph Seilacher, came around and he created essentially an encyclopedic version of ichnology, where he went around and classified everything into a bunch of broad groups. This is a picture of him in the field. He died just a couple of years ago. But here are his behavioral classifications. Rapicnia, moving traces. Pasicnia, grazing traces that are feeding and moving at the same time, kind of like cows. Agrichnia, farming traces, comes from agriculture. Phononychnia, feeding burrows. Domicnia, dwelling, dome meaning home. Fugichnia, meaning escape traces, like fugitive, escaping. And then cubichnia, resting traces, like Rusophycus, the one we looked at, that would have been a cubichnia. All right, you never have to say those names out loud. Don't worry, never going to be part of your exam. Neither is spelling. So here are some examples of it. Here are our fugichnias, escape traces, our resting traces, or home traces, looking for food, farming traces, all kinds of things here. So one of the cool things I want to point out here is you notice these farming traces. Do you see how none of the burrows or anything touch each other? Do you see how there's never a point where they track over each other? That's actually something we see happening across the Ediacaran to Cambrian boundary too. So at first, the burrows we find actually did go back and forth over each other, meaning organisms were going over the same places looking for food, even though it probably wasn't going to be there. They already checked that place. But somewhere along that transition, we start to see higher level thinking occurring, where animals are starting to say, okay, let me make a pattern. We're going to search this more systematically, which I think is so cool. So what we can do with ichnofossils is actually really cool. We can take groups of them and start to reconstruct really specific paleoenvironments, like water level, water energy, and things like that. Now, Ichnofacies zones are named after ichnofossils that are characteristic. So for example, it's kind of like biozones. We can name it after specific animals. So even though that animal may not be found in that particular section, it typically is. Does that make sense? So we'll go through a couple of them. There's a whole bunch. We're only going to go through a few of them, just what they mean. So this is what they kind of correspond to. We have the deep water ones here, starting with Nereites, that's the name of it. Then we have one a little bit more shallow, Zoophycos, Cruziana, Skolithos, and then Glossofungites and Tryptonites. These are typically associated with hard borings and things like that. So we're going to go through and we're going to start in the deeper water first with Nereites. There's a bunch of different trace fossils that could potentially be there. So Nereites is here. This is one of the characteristic ones, of course, it's one it's named after. It's a meandering trace. Do you see? Meandering means not really walking with a purpose, kind of going in and out and walking around. And you can see how that might happen here. You see this trace, it kind of doesn't have a purpose, not like something like number 4 Paleodictyon here. That very clearly has a nice little pattern. Here is our Paleodictyon, looks like a honeycomb, right? Absolutely beautiful to look at. So there is some discrepancy here. Some folks call Paleodictyon more of an agriculture trace. Some call it more of a dwelling trace. Either way, super cool to look at. I kind of tend to lean towards more of the agriculture farming one, but I don't know for sure. So Nereites here, again, it's systematic mining of the top layer of sediment for food in deep water. They're often associated with turbidite deposits. They're not typically incredibly diverse. There's usually only a few different types of trace fossils here. Okay, so Zoophycus, also found in many depths, typically lower oxygen water, typically deeper water as well. Now, most traces are complex feeding and deep burrows. What this means is the organisms there are kind of living in a rough life. There's not a whole lot going on. So if there's deep burrows and complex feeding traces, they're looking for food actively. It's not right there to find. So here is a Zoophycus. This is from a friend of mine, Adrian Lamb. This was found in a core about 1,500 meters in depth, made by probably a worm or something. So again, low oxygen is typically going to correlate with lower diversity of trace fossil structures. Cruziana. We can see that the diversity is picking back up. We've got a whole bunch of things here. Chondrites, Tychicnus, Roselia, Planolites, all these different types of traces happening. This is below the fair weather wave base, meaning it's not really being affected by normal wave activity. Nice conditions, high diversity of traces, high behaviors, likely high oxygen as well. Here is our Thalassinoides, super beautiful. All right, so moving on up, we're both still back. Ugh, words here, huh? We're still in Cruziana. All right, so they use the substrate as both shelter and food, very diverse assemblage, lots of nutrient delivery is coming in a lot. It's typically finer grained substrate than Skolithos, which is our next one. And that typically is going to mean that Skolithos is higher energy. Remember, coarse grain goes with higher energy. All right, so Skolithos. Shifting sands, higher energy, intertidal or emergency conditions following turbidites. Turbidites are storm deposits, and they lay down in 5 specific layers. The last one is the first series of those layers is typically the largest stuff that settles down. We know that based on the principles of settling particles. So here is Skolithos. Again, because it's low diversity, we can assume that it was a pretty rough place to live. So if the environment is continuously changing, there's not a whole lot of organisms that super love dealing with that. So lots of vertical burrows so that they can stay away from the shifting sands and all of the craziness going on above. Lower diversity, this time not necessarily because of low oxygen, but because of changing environment. Most of these are going to be resting traces or escape traces. Now these two, Glossofungites and Trypanites, these occur on hard substrates and they're typically independent of depth. Some will only happen on rocky coasts, but for the most part they're independent of depth. So these two could be existing on other organisms, like a boring into another organism. There's a bunch of animals that make these, and really what they tell us is that there was a hard or firm surface. So things like echinoid borings or burrows into other organisms, things like that are going to be what we see here. So for example, if you pull out your fossil kit real quick, you probably have been wondering, hopefully you figured it out by now, but you've probably been wondering what that weird, long, tubey looking fossil is in the front. Hopefully you figured it out because it's a bivalve, and that's one of the ones you were looking at during our last lecture. All right, that is a shipworm. Here it is in life. And it just burrows straight into wood. This is one of our examples of a hard substrate. All right, so this is a piece of wood. This is Teretolites. The trace is named Teretolites. This is not Teretolites, right? Remember, that's a trace fossil name, not the organism itself, even though this is one of the ones that we know makes that trace. So one of the things we can use for this is when the ichnofacies change quickly, it can indicate that there was a change in environment. So something's happened super quickly around here. We have mud, sand, and mud. Excellent. We also see some other trace fossils here. Sedimentary structure. Can you recognize this sedimentary structure? It looks like cross stratification, right? But it looks a little weird. They're big cross stratifications. Well, that's hummocky cross stratification. And if you remember from your sedimentary petrology lectures, hummocky cross-stratification is typically associated with larger storm events. So it's below fair weather wave base, but not below storm weather wave base. So we can tell that this may have been like a bad storm. There might be some turbidites here, some escape burrows might have been occurring, like in 13 here with the Skolithos trace fossils, something like that. So there you go. So this could have been a storm event on the mid-shelf. We can also, let's try this one. Let's say we suspect shallower water ichnofacies. Maybe Cruziana, potentially Skolithos, depending on. We can look at sedimentary structures to kind of support or reject. Based on this picture here, would you support or reject? What are these? Well, these are ripple marks, right? And they look fairly symmetrical, so this is probably an ocean. Excellent. I would support that hypothesis, right? These are typically only going to form by the fair weather wave base, which means it's probably pretty shallow marine water. Could be Skolithos. We don't have any fossils to say yes or no. I've made the scenario up in my head. Now, what would you look for if you suspected low oxygen environment? Not necessarily with your fossil assemblage, we just talked about that, but what else might you look for? Could the color of your mudstone help you? If you were looking for low oxygen, could you look for mineral associations like pyrite perhaps? There could be a couple lines of evidence to look for. So here we have where the energy level that particular bed forms like ripples or dunes, antidunes, etc. will form. So if we know that we've got ripple marks, that means that our fossils had to have been forming in water about this range. All right, hopefully you remember this chart from sedimentary petrology. So one issue with ichnofossils is what we're going to call time averaging, tiering and time averaging. What this means is that animals who burrow are kind of rude. They really don't care who was there first, and they really don't care about the paleontologists who might be studying those burrows a little bit later. They'll build right on over other burrows, or they will move them around so they're not directly competing. That's something called niche partitioning, trying to increase resources to decrease competition. If we see deep tiering, that typically indicates, meaning lots of different things, that probably going to indicate well oxygenated bottom waters. But as that sediment builds up and tiering continues to happen, it could be really difficult, making distinct structures harder to see. For example, let me go back one. You can see that these distinct structures are getting harder to see. So that's one issue with ichnofossils, is time averaging. The amount of bioturbation, or Ichnofacies indexes, can tell us a lot about bioturbation. How much has the sediment been messed up? That can tell us a lot about biodiversity and the general health of the community at that time. So here we have increasing bioturbation, more diversity, or it could mean higher population sizes, but typically both. Now, what else can we learn about trace fossils? So much! Think about a body fossil. We find a dinosaur. Boring. It's just bones, right? No, I'm kidding. That would be like the highlight of my life, right? Okay. So, body fossils can tell us a lot, but trace fossils can tell us so much about the behavior of an organism. It can tell us who is being predated on. It can tell us orientation of currents. It can tell us so much. So here is some coprolite. You'll notice it is a lovely spiral shape. This is likely a fish poo. So what we can tell from this, sometimes we can tell the shape of intestines, right? There are some animals that only poo in cube shapes. Why? No clue, but glad we have them today, because if we find cube-shaped poo in the fossil record, we'll have a good idea of who made it. We can find out diet. What kinds of bacteria are in your poo can tell us a lot about what you ate while you were alive. It can tell us about the parasites that lived in your system, if you died from parasites, or if you just have the parasites that live in your system always, and they're kind of in a symbiotic relationship with you. Who knew fossil poo was so cool? Maybe it can tell us about bathroom habits. This is one of my particular favorite trace fossils. This is a Urolite, yes, a urine trace fossil from a sauropod dinosaur. So someone took it upon themselves to draw this here. I actually pulled this off a scientist's page, I promise you. So, and this is a photo here. I left the Twitter feed down here so you could see it forever. It's pretty great. But this is a urine trace fossil. Now, sauropod dinosaurs were pretty big. These organisms probably went to the bathroom with a little bit of gusto, if you're getting my drift. So how much energy would it take to pick up and move those sand grains? These are the questions you never thought you'd have to answer, but here we are. Okay. I bet my mom is super proud of me. So here's the Holstorm diagram of the size of the grain and the energy required to move it. So if we have sand grains, we know the approximate velocity of that fluid that's going to have to move those sand grains. The more you know, folks. Now, does that mean we could look at modern animals and we could see how animals in the past might have been making bathroom traces? Because you can imagine, if you just saw something like this, your brain might not have gone to pee spot, right? Exactly. So someone wrote this paper. This is a 100% real paper written in 2003. This is a 100% real diagram that I took from a peer-reviewed scientific paper from a professor with a PhD, all right? The penguin. How much rectal pressure would be necessary to expel fecal material over a distance of 40 centimeters? Physics can answer that question for you, the question you never know you needed an answer to. But this might help us understand what penguin feces marks might look like in the fossil record. Mating rituals. Now, mating rituals you would think could not be preserved. But what if they could be? So we're going to watch this video here. And I'm going to mute it because we don't need to hear the sound to it, but it is David Attenborough, so I would Isn't that beautiful? Absolutely gorgeous. So cool. I'm going to stop because I sound like the B-rate David Attenborough. And there we go, from far way away. Probably C-rate David Attenborough, to be honest. All right. So, we recently found evidence to suggest that dinosaurs had similar mating structures as birds. So some birds will do weird little dances where they scratch around their nests. And we recently found something that matched that, but in theropod dinosaurs, large theropod dinosaurs, because birds, of course, today are theropod dinosaurs. So here is the artistic rendition that went with that paper. Do with it what you will, right? There you go. But these are traces left behind, so we think that dinosaurs probably had very active mating rituals just like birds do today. Could we find vomit traces? Again, the questions that we all needed to know in life. Here is that diagram from the first image, or from the first slide here. This is from Anthony Martin's Dinosaurs Without Bones. It is an entire book on trace fossils and dinosaurs, which is where a lot of my material came from today. Highly recommend. So, I know you were thinking it at some point in your life, what would happen if a sauropod puked, right? We can calculate using physics. We know that the neck is approximately 45 feet off the ground. We know gravity is 9.8 meters per second squared. So if an organism were to vomit, the impact would hit at approximately 68,000 newtons, which would, in fact, probably kill these wee little itty-bitty velociraptors here at the bottom. Horrifying, right? But awesome. We have not found traces like this, unfortunately, but there's still a lot of fossils to be found. We can see predation. This is a giant tortoise shell with crocodile bite marks in it. There's no healing marks on the shell. This might indicate, and likely does indicate, that the tortoise did not survive the encounter. On the other hand here is a snail that fought back against a crab. These are from Christina Barclay. This is her research. A crab managed to yank off an entire whorl of this snail shell. But one of the things you can see are healing marks on the shell right here. That's actually new shell material covering it, which indicates that the snail fought back and survived, which is so cool. This is what a typical crab does. It actually just takes a huge chunk out of it with its claw. Not sure why it decided to take the whole whorl, but it did not work. That snail survived. Really cool. Can we tell familial relationships? These are the Laetoli footprints, some of the most incredible and beautiful fossils that show our deep roots in humanity. These are Australopithecus afarensis fossils, so early humans, approximately 4 million years ago. Some people would argue that they might not be fully human, but they are some of the ancestors that we recognize very close to our lineage. All right? So what we see are 2 adults and a child. or a juvenile event of some kind, walking. So could that tell us a little bit about familial relationships? It can also tell us about speed. Based on the length of the footprint, it can tell us about how tall the organisms were, which is absolutely cool to think about. It can also tell us about weight distribution. Here are some models about how the Laetoli organisms would have walked, and where they put more pressure. And they actually put more pressure on the outsides of their big toes than humans do today, meaning they likely walked more like a chimp or a gorilla, but there was definitely still some bipedal motion, meaning they were also very similar to humans. Here are your review questions, and I will see you guys